So thanks everybody for joining us this morning. I'm Lynne Carpenter-Boggs. I'm in the Cropland Soil Science Department in Pullman, and mostly a soil scientist with emphases in the fertility and soil biology areas and systems studies and sustainability and organic agriculture. So, the last few slides in here also will be from Dr. Maron Friesen, so you'll hear from her. And yeah, I've been doing some projects with compost tea for quite a few years now. And of course, we have just a little bit of time to tell you what we know about compost teas and this new project that Carol mentioned. So let's start out with some real basics, if my slides will move. There we go. All right. So just definitions to start out with. Compost tea can be extremely simple, just compost and water. Compost teas can also contain any number of microbiological inoculants. They often will include sugars or other sources of food for microorganisms. They can have plant nutrients such as rock powders or whey powder or other nutrient sources added. And then compost tea is fermented anywhere from an hour to a week. That can be static, meaning non-aerated, or in an aerated state. I'll mention that if you have compost and water, including any of these other additives, and it's not held or fermented for at least an hour, it's not actually considered a compost tea legally. It's just called a compost extract. So there is some brewing time. So this process primarily extracts the nutrients and some of the humic acids from the initial material, and it also dislodges the initial microorganisms in the compost. And then depending on the nutrients and temperature, etc., it can also increase the microbial population. All right, so the two main ways are just doing a passive steeping versus brewing or aerating process. So, and either one is considered a compost tea, and depending on who you ask and what you're trying to do, you'll find both of these methods. It can be super simple, putting compost into a bag or a sock and setting it into water. Of course, that's going to be a cheap way to do it. If it sits more than a couple of hours, it is likely to go anaerobic, and after a day or more, those are likely to start producing off odors. Not that that's necessarily a bad outcome as far as the nutrient content of the tea, but just something to be aware of. If it's aerated, you can do, again, very simple things like using aquarium bubble stones. You can buy specific blenders for either a bucket size or up to thousands of gallons. This clearly will provide more consistent aeration and an aerobic environment for the microorganisms. And you can spend any amount of money that you would want to spend on various sized brewers, or you can make your own. So those are the two main ways, though. And then all sorts of additives have been used. Just a few would be kelp extracts, humic acids, rock dusts, molasses or other sugars, whey powder, dried herbs, or even commercial blends of inoculants, and who knows what in the commercial compost tea blends. So I want to mention that if molasses or other sugars are added, those definitely are quick food sources for the growth of bacteria, but those bacteria can include human pathogens. And there are increasing regulations around the use of sugars and including molasses in compost teas. because of the potential to actually grow up harmful microorganisms. So, one of the reasons people are interested in these teas is that we do increase the population of microorganisms in that solution. So generally speaking, when we provide microorganisms a good environment that includes food, warmth, and give them some time, that's going to grow more microorganisms. So we increase the population. But I want to point out that microorganisms are very diverse. And so certain foods or certain conditions will favor different microorganisms. So depending on what foods we give them, we might culture up a very particular portion. It's called a recruitment process. We brew up just what might be a small population to begin with, but if that's who we're favoring, who we're feeding, then we can have a big community shift in the microorganisms we end up with. And so some of the work that we've been doing with compost teas is kind of looking at, well, what's the relative effect of trying to use different composts versus using different compost tea recipes, like different amounts of aeration, different nutrient additives. And what we've found is overall, it depends a whole lot on these other additives, much more so generally than which compost you use. So for instance, this is just looking at the electrical conductivity, but we've looked at lots of chemical and biological factors, and you can see that there can be pretty consistent results from batch to batch using a same recipe. So for instance, when you look at this compost tea recipe A to the far left, very low electrical conductivity. And then the next recipe, we do 3 batches of these, and all of those batches are pretty similar within the same recipe, but dramatically different conductivity than the other recipe. So, we've been practicing basically, like, can we make a fairly consistent tea? That's one of the things that we really need to be able to do to be able to do research on these kind of things. So some of the reasons people are interested in using compost teas or studying them is that there's potential for them to enhance plant health, provide plant nutrients, speed up decomposition, suppress plant diseases, and because of those things, to reduce fungicide, other pesticide, and fertilizer requirements. And so I'll touch on just a little bit of that research that's happened. And of course, research into teas is continuing to expand. So just a couple that have looked at the fertilizer value of compost teas. One done on pak choi showed that the above ground biomass, the harvestable portion of the plant was increased 20% with the use of compost tea. And but that also gave fairly similar results to using a soluble fertilizer, although increased growth compared to using just a chicken manure compost with the same nutrients. Another study on strawberries showed that this was using a non-aerated tea, so just a passive steeping, but it's provided the same level of micronutrients and growth as compost or fertilizer. So people have been just comparing these two other nutrient sources and finding out that it's often a similar result. Completely different side, though, there's the microorganisms. And so compost tea can be used or thought of as a microbial inoculant onto a plant. And plants do have microorganisms on their leaves, what's called the phyllosphere, or on their roots, and what we more commonly know this term of the rhizosphere. And plants also have microorganisms often inside their roots and stems and leaves. And this is a whole area of study of endophytes, microorganisms in plants that could be affected by something like compost teas. So I want to touch on the idea of using compost teas as a disease suppressant, and there's a whole like legal statement that we need to cover first. So anytime anybody tells you about something that's controlling a disease, you need to think of this as a pesticide. So as researchers, we're allowed to do a lot of things that we're just allowed to do them under an experimental use permit or, as experiments. Once we move things out into the field or like start telling people that, hey, this might work to reduce disease, it's a completely different story legally. So please, no one go out and say, oh, Dr. Carpenter-Boggs said this reduces pythium. No, I'm not saying any of that. So, and nobody can tell you that that's correct either. So anything that is being used to treat a disease is a pesticide and has to be registered with the EPA. So legalese there. All right. But so there are more and more studies about compost teas and their effects on plant diseases. But the results are mixed. So there are a number of studies out there showing compost teas can suppress plant diseases. Sometimes you see results that say the diseases are worse, though. And sometimes they enhance plant growth. And once in a while, you see studies that show they've actually reduced the harvestable yields too. And sometimes there's just no effect. So it's not a completely reliable material yet. And one of the reasons for that is that these compost teas are all different. And that's why we've really tried to kind of narrow down on some recipes that we can rely on. So, but given all of that, the results in the literature are mixed. Here's a list of some of the plant diseases that have been reduced in at least one published study. So there's many different crops and many different diseases that, at least one study has shown they have reduced these diseases. And again, I'm not recommending these materials for disease control, just saying that the studies are happening. So studies have shown that compost tea can be a valuable nutritional supplement to plants, although it is a fairly weak supplement, fairly low nutrient content. Sometimes they do seem to stimulate plant defenses. We always say we would love to see more peer-reviewed research, and we are starting in on a new project to do that. If you're going to use compost teas, please check with your current rules for your system and crops. Check with any certifiers you're working with. There are rules about using compost teas, especially with fresh produce and organic certified crops. So as Carol mentioned, we have a new project that you're now a part of, whether you expect it to be or not. This is actually part of it. We really want to better understand grower needs and your research questions about compost tea, so we can start pursuing those. And another of our short-term goals is to better characterize these microbial communities in teas and their effects on residue decomposition, which we have already heard is a particular interest. So here to talk a bit more about the microbiological side of the study is Dr. Maron Friesen.
Thanks so much, Lynn. Hi, everyone. Thanks so much for joining us this morning. My name is Maron Friesen, and I am an associate professor in the Department of Plant Pathology at Washington State University in Pullman. And although I'm in the Department of Plant Pathology, my true passion is actually beneficial microbes. And so I'm really excited to get this project launched with Lynne on some of the beneficial effects that microbes can have. And so my lab does a lot of different approaches, but one of our primary tools in understanding microbial communities is through high throughput sequencing. And, that lets us look at sort of who is there in terms of the microbiome of the tea. And we also have some specific tools like quantitative PCR that we've been using and developing in order to look at particular functions that different microbes can perform, either in teas or in roots and rhizospheres. And so, yeah, just wanted to throw out there that if you are brewing compost tea, we would love to sequence it just as a exploratory part of this initial project that we're launching. And so for this project, so we have another slide, I think. Yeah, so my role in this project will be helping out with characterizing these tea microbiomes. And so we will be looking both at the structure of the microbiomes, so the sort of who is there. So we are planning to extract DNA from a series of lab-grown compost teas that Lynne Carpenter-Boggs, using recipes that she has experience with. And then we will sequence the bacteria and fungi present and analyze those communities. And we also are really interested in the different functions of these teas. And so there's lots of different potential functions that these teas could have, but for the purposes of this current project, which is quite a small project, we'll be focusing on residue decomposition. And so our plan is to add these different teas to residue in jars with field soil, and then incubate those on the timescale of weeks to months and actually measure the decomposition rates. So trying to just generate some ideas about the potential that these different teas have in terms of decomposition. And then kind of at the end, once we have data on the structure and the function of these different teas, then we will compare those two sets of data and see if we can come up with hypotheses about if there are particular microbes or groups of microbes that might be contributing to this function of residue decomposition. And I think with that, we will transition into the Q&A.
Great. Thanks so much. What a wealth of information.